Today, we're going to talk about a tiny molecular machine that is so clever, so ingenious, it's basically been called a biological mousetrap. And the craziest part? Its entire story is written deep inside our own DNA.
So think about this for a second. Your body is just packed with these super powerful enzymes called proteases. You can imagine them as little molecular scissors, and they're essential for, everything from digesting your food to fighting off infections. But here's the catch. If you just let them run wild, those same scissors could easily start shredding your own healthy tissues. So the big question is, how does your body keep these incredibly dangerous tools on a tight leash?
And believe me, the stakes could not be higher. It is an incredibly delicate balancing act. I mean, look at this. This balance is at the absolute heart of blood clotting, our immune response, you name it. One little slip-up, just one enzyme going rogue, and the whole system could be thrown into complete chaos. Getting this right is, well, it's fundamental to staying alive. To solve this huge problem, evolution came up with a solution that is just breathtakingly clever. It's not a leash at all. No, it's a trap. A molecular mousetrap that we call the serpent.
Now, a serpent isn't your typical inhibitor that just kind of gets in the way for a little while. Oh no, it's what we call a suicide inhibitor. And that means its mission is strictly a one-way trip. It is designed to find its target protease, neutralize it for good, and get destroyed in the process. It's really the ultimate act of molecular self-sacrifice.
Okay, so let's get a look at the trap itself. This beautiful, complex thing you're seeing is a serpent, and it's in its high-energy, set state. You see that little red loop kind of sticking out? That's called the reactive center loop, or RCL for short. And that, my friends, is the cheese on the mousetrap. It's perfectly designed to lure in a target protease for what it thinks is going to be an easy meal.
And this is where the magic happens. The protease takes the bait. It cuts that loop, but the instant it does, snap. The serpent undergoes this massive, unbelievably fast change in its shape. The trap is sprung. It drags the protease clear across the molecule, which is a huge distance at this tiny scale. And in the process, the protease is literally crushed. Its active site is just mangled beyond repair. The whole thing, the serpent and the protease, is now totally inert, and the cell just cleans it up. Job done. Incredible.
So we have this absolutely amazing molecular machine. But you have to wonder, right? Where did it come from? How did it evolve? To answer that, scientists had to become genomic detectives. And let me tell you, this was no easy task. I mean, there are thousands of different serpents out there, in animals, in plants, even in some bacteria. Just trying to compare how they look on the surface wasn't going to cut it for building a reliable family tree. The relationships were just too complex, way too ancient. Researchers needed a much better way to read their history.
The big breakthrough came when they decided to look at their genetic blueprint. So, instead of just comparing the final proteins, the researchers analyzed the underlying gene architecture. That's the pattern of the coding and non-coding parts of a gene. And what they found revealed this hidden order. It was pretty remarkable. They found that all the serpents and vertebrates, like us, could be sorted into just 6 fundamental designs. 6 groups.
And this new classification system, well, it unlocked a story of evolution that stretches back over 500 million years.
What the genetic blueprint showed was, it was just astonishing. All 6 of these core serpent designs were already there, already in place, before the great split between fish and land animals, which happened over 500 million years ago. That means this fundamental toolkit of molecular mousetraps is incredibly ancient. It's a system that evolution perfected really early on and has just stuck with ever since.
And what's really cool is that this research uncovered 2 very different evolutionary stories playing out at the exact same time. On one hand, you've got the innovators, serpent groups V1 and V2. These guys evolved really rapidly in mammals, diversifying to fight off fast-changing threats like viruses. But on the other hand, you have the old guard, groups V3 through V6. These were deeply conserved. They've been performing the same core jobs for 500 million years, machinery that was just too important to mess with.
And the history goes even deeper than that. I mean, get ready for this. The origin of one specific serpent in your body, neuroserpent, which protects your neurons, can be traced back more than 650 million years to a common ancestor we share with sea anemones. Just let that sink in for a second. A piece of the molecular machinery in your brain is an echo of a simple little animal that lived in the oceans long before the dinosaurs even existed. So, what's the big takeaway here? What does all this mean?
It means that our own DNA is a living history book, and the serpents, they're one of its most fascinating chapters.
You know, a line from the research really sums it all up perfectly. It says that our bodies, and the bodies of all vertebrates for that matter, they all run on a core set of these ancient, highly conserved molecular machines. We have inherited a proven 500-million-year-old toolkit for survival, a collection of mousetraps that still protects us every single second of every single day.
It really makes you wonder, doesn't it? If a mechanism as elegant and as critical as the molecular mousetrap has been hiding in plain sight inside our genome all this time, what other ancient stories, what other ingenious solutions are still waiting in there to be read?